Much for that kind introduction. Thanks for inviting me to talk. This indeed has been a journey. It's a journey was probably 5 years in the making before we finally made it to the clinical lab, and I hope you can appreciate some of the findings that aren't just all based on science too. Here are my disclosures. So I have a couple learning objectives for this talk. I hope to give you an understanding of how you can use immunoglobulin light chain mass distributions to reveal plasma cell disorders. I'm going to describe some of the challenges when you face something as old as immunofixation, which is a gold standard assay. And I'm going to give you some cases just to show you why we decided to move to the new method away from this gold standard. But to get everybody on board, I just need to define these plasma cell disorders a little bit in the lab. The defining feature is something we call the M protein, which really stands for, it used to stand for myeloma-associated protein. Now it's sort of morphed into monoclonal protein. And it's a very unique target in terms of clinical chemistry in my mind, because it's the presence of a monoclonal immunoglobulin expressed above the polyclonal background in the serum of the patient. There aren't too many targets that we go after in clinical chemistry where it depends on some other, background immunoglobulins in there, because in reality, every immunoglobulin is monoclonal. It's just we're looking for those that are overexpressed. So the current methods that are been around since the 1960s, actually here at Mayo, capillary electrophoresis or gel electrophoresis. We've always used gel here at Mayo, where you put the patient's serum on this agarose gel, you add electrical current, and then you stain the proteins using a dye, and then you run a scanner across. And in the case of plasma cell disorders, we're looking for the immunoglobulin region here in the gamma region, and something that's overexpressed like this shows up as a spike. We call it an M spike, and that's how these plasma cell disorders are mostly detected in the clinic. It's been a great tool. It's the only quantitative tool for defining the amount of M spike. It's relatively inexpensive compared to other tests. And its role in detecting these plasma cell diseases, really, you can find them. It's for diagnosing and monitoring them over time, because the M spike is known to be directly proportional to the plasma cell burden in the patient. It has some problems. It lacks specificity. There are artifacts that you can get on a gel. Things like, if you don't get completely clotted serum, fibrinogen can interfere. Hemolysis and LDL particles can also go into the gamma region. Rheumatoid factor can sometimes glom up immunoglobulins on the gel, make it look like an M spike. IgG4 disease is a newly appreciated one where IgG4 will get overexpressed, and it has a relatively restricted migration on gel electrophoresis. And it can only quantitate so far. As I tell people, the gel can only get so blue, and so it has limitations at the upper end and has limitations on the lower end. Below 1 gram per deciliter, you kind of have the interference of the polyclonal background. And at higher ends, you have this dye saturation, which is a problem. So then it also has some limited clinical sensitivity, because not all M proteins migrate in the gamma region. If they happen to migrate in this beta region, sometimes you can't see it without further testing. All right, so to overcome that, immunofixation was invented. We've been running this same gel in our lab since 1980. Here is another agarose gel. You put the patient on here 6 times. You do the electrophoresis, and you stain all the proteins, or you fix all the proteins to the lab in the upper one. Here you use anti-IgG, anti-IgA, anti-IgM, anti-kappa, and lambda, and then you're trying to isotype these M proteins. It has very high specificity because you know for sure it's an immunoglobulin. It does have higher clinical sensitivity because it can see things under the beta region. It gives you isotype information, which is important to the clinicians to try to look for the disease, as IgGs and IgAs tend to be myeloma, where the M's tend to be LPL lymphoma. And so its role, again, is also in diagnosing and monitoring. It's higher cost compared to serum protein electrophoresis. So sometimes it's used in a reflex mode instead of just as a screening mode. And the downside is it's not quantitative, so you end up having to do both. So what kind of disorders are we talking about? And this was what makes some of these diagnoses challenging. Of course, there's frank malignancies of plasma cells, of which the dreaded one in my mind is multiple myeloma, very bad disease, but also Waldenstrom's macroglobulinemia. You can have malignant plasmacytomas and LPL lymphomas. But you also have a host, and these are the majority of the patients you'll see where they have no symptoms. And so we consider these pre-malignant, like MUGUS, where the patient has a monoclonal protein, but they have no symptoms from it. And this makes up the highest percentage of the patient population you're detecting in the lab, because it's estimated that if you're over the age of 50, 3% of us would have a monoclonal protein. But there's also these diseases of plasma cells where it's not really the cells that are malignant, but what the cells are making are malignant to the body. Things like AL amyloidosis, where you make a small amount of these M proteins and they precipitate in plaques in your organs and cause you organ damage. Light chain deposition is another one, and things like cryoglobulin and POEM syndrome. And the challenge here is that these usually present, these diseases on the left present with very large M spikes, but these on the right tend to have very low levels. So you need to detect, if you're trying to rule out all of these, you need to detect high level and low level M proteins. So why look for newer methods? This has served us here at Mayo Clinic since 1960. Why look for newer methods? And this is one of the things I talk about is I was on part of the CAP Diagnostic Immunology Resource Committee. And a lot of the societies for oncology recommend that if you really want to rule out one of these plasma cell disorders, you should do what's here in the orange. You should do a serum protein electrophoresis, an immunofixation, and measure the free light chains in a patient. So we sent this question out and we sent it to 741 labs, and we were wondering, okay, well, what does physicians use in your laboratory to find one of these plasma cell disorders? And only 6% of the labs are actually using the recommended screening protocol. And I can kind of understand why. If you look at it, most people were running a serum protein electrophoresis. If they didn't see anything, you know, they would then call it negative. But if they saw something, they'd send it over to immunofixation. But a lot of people just use SPEP. You know, and we've experienced that too. This IFE is pretty labor intensive. It's got a low throughput. And when I talk to some of the colleagues that do this, say, you know, we just don't have enough resources to do IFE on everybody. So from here now, I'm going to move off into a story of how mass spec sort of came to attention into our lab. I'm going to start with a case. This was really the first case we ran by mass spectrometry in our lab, and it was helpful in the diagnosis of this individual. So this was a 54-year-old male. He had limited prior medical history. He came in from living in the woods, so we called him the man in the woods. He had a lot of symptoms. He was just basically sick. He had a temperature, swollen lymph nodes, and back pains. And then some of his lab findings, he had elevated liver enzymes, but they noted this elevated total protein. So someone got suspicious for this M protein and sent it to us in the lab. And this is what his serum protein electrophoresis looked like. So again, here's the albumin. Here's the region we're looking for an M protein. Well, you kind of see this pseudo restriction. We see these quite often by SPEP. But this is quite large. You can see the gamma region was 4.6 grams per deciliter. And although it's possible to get a polyclonal response up that high, it's somewhat unusual. You can see his total protein is elevated. So then we did immunofixation on him, and to clear this sort of artifact, we reduced the immunoglobulins with DTT. We do the immunofixation, and some of the lab were questioning, is this really restricted in the gamma region? But it looks kind of broad in kappa. So we were having a hard time trying to figure out. So we also ran the free light chains on this fella, and he had an abnormal ratio, which would suggest there's a monoclonal process somewhere in here. So this is where the differential diagnosis comes in on this patient. If I gate this big spike, I'm going to give him a diagnosis of multiple myeloma, or a presumed diagnosis of multiple myeloma. He's going to get a bone marrow biopsy worked up fairly extensively. If I don't gate it, I'm going to assume this is a polyclonal hypergamma patient, maybe due to autoimmune disease or liver failure. But this is kind of a high level to get to this kind of diagnosis. And, you know, does this patient also have a cryoglobulin? Because we were having problems getting his serum into the gel. So now we're going to go back. So before that case even happened, 2 years prior, the hot topic in myeloma was a serum MRD marker in myeloma. And so we started out trying to use the M protein as one of these high sensitivity, minimal residual disease markers in myeloma. And the idea was to use traditional shotgun proteomics. So if you take an antibody or an immunoglobulin like this, you have this variable region, which is the target for the antigen. And this target area, the variable region, has a very unique set of protein sequence in it. So the idea was if we could sequence that variable region, then chop that sequence up with trypsin, like normal shotgun proteomics, maybe we could find a peptide in there that we could follow below the polyclonal background to look for residual M protein in that patient. And so we were using high resolution mass spectrometry to do this. And so you have to remember that, you know, there's quite a few of these immunoglobulins that are possible in the immune system. B cells are very unique in that they rearrange their DNA to produce one of these antibodies. And if you go through the traditional thing that you're taught in immunology, you come up with this idea that there's a possibility of 2 times 10 to the 12 different antibodies that could be made. And how many of these are in serum and circulating, we don't really know, but that made it really challenging. But this is really why, you know, you have this gamma region. The gamma region's naturally broad because the variable regions are changing the charge of the immunoglobulin slightly, and they migrate broadly on this gel. So part of me said, well, wow, this is like, if you have this many, this is like trying to find a needle in a haystack. Could it really be done or not? So we spent a lot of time doing this back in 2014. We took plasma cells, we sequenced the variable gene regions out of the heavy chain in some of these patients. We created the protein, the light chain and heavy chain proteins. We did a theoretical trypsin digestion on our sequences. We picked out peptides, and sure enough, we were able to get down to sensitivity somewhere in the neighborhood of 1 milligram per liter of serum. So it was very successful. We were very excited about that. And this just shows you what modern mass spectrometry can do. So this is just an example from one patient. So we've isolated this patient's IgG repertoire. We've digested it with trypsin, and the top shows just the chromatogram that's coming out. So we can see that ions are being formed, and these immunoglobulin peptides tend to come out between 4 minutes and 10 minutes. And so you can go in there with these modern mass spectrometers and do time of flight measurements and fragmentation measurements on all these peptides. And down here in the left corner are all the different peptides. Each dot is a peptide that we detected, and the one in red was the one from the variable region. So it just shows you what's possible with some of these modern mass spectrometers. And so this is very interesting and was a very promising way to start to look for M proteins at very low levels. But I said, okay, you found it. And it says, congratulations, it only took you 95,298 seconds. And that was part of the problem of trying to implement this. So we had to sequence the plasma cells. We had to create these structures. We had to augment databases with the peptides. It was a very labor intensive process. And we found some real limitations to this too. That for us, we wanted to find a unique peptide for each patient from the variable region. And in 20 patients that we did this on, we couldn't find a unique peptide in 2 of the patients. And remember that although that's a variable region, there is framework regions within there. And if you get peptides across the framework region, it's very hard to find unique peptides. This would never be used to screen for M proteins. Then we kept wondering, what are we going to do for internal standards for each patient? So if Mr. Smith is here, do we have to create a labeled peptide for Mr. Smith? That didn't seem feasible to us. And then we asked ourselves, well, how unique is each one of these peptides? And this is a very interesting study done by a group in the Netherlands, where they just took normal healthy donors from the blood bank. So they isolated all their IgG. They added an IDES enzyme, which cleaves off the FC portion. They purified the FAB portion, and then they did trypsin digestion of it. They got the peptides and did traditional shotgun proteomics. And they said, well, how many total number of unique peptides did they get from an individual? So it was around 41,000, right? And so this tells me that we're probably in the millions of different immunoglobulins in the patient's serum. Not 100% sure of that, but at least we're not to the 10 to the 22nd. They repeated this in 7 healthy donors. And the percent of peptides unique to an individual, only 37 peptides were found that were unique to an individual. And how many peptides were shared by all individuals? Well, it was 83%, which told me there's a lot of redundancy in using these gene regions to select these, and that we might be in trouble getting unique peptides when we got down below the polyclonal background. So we decided to change our approach. So what we decided to do was to, instead of digesting it, just to do sort of a top-down proteomics approach, where we decided to denature the immunoglobulin and break it into its heavy chain and light chains. And the reason we did that is because this oligosaccharide on the heavy chain is not clonal in monoclonal patients. You get various amounts of glycosylation on that heavy chain, and it makes it very hard to use for a mass marker. So we decided this time we would go after the light chain. And so this is the basic paradigm behind the test. So we take a plasma cell, makes a unique immunoglobulin, you break it into its heavy and light chain, and then you measure the masses. And so what I show here, black is the heavy chain, right? And so you'll get a different glycoforms. And if you have a spectrometer that can resolve it, you can see those glycoforms. But for the most part, and I'll show you later, this is an oyster, you get one mass for the light chain. And so we're going to use that mass as the marker of the clone and the intensity as the abundance of the clone, just like you would in SPEP. So when you do that on normal human serum and you use a higher resolution mass spectrometer, you actually get two distributions, which should have been obvious to us. Wasn't obvious at the first part, but because of the difference in the constant region, the mass of the kappa and lambda light chains will segregate. But if you have somebody with a monoclonal protein, you can kind of see the kind of resolution that you get. So this up here would be the patient's M protein. But the resolution also has a downside because you can see PTMs on this. And what we know now is immunoglobulin light chains get non-enzymatically glycosylated, just like hemoglobin does. So you always have to count for these plus 160 sometimes in these patients. So we did this. We have this method now where we purify the IgGs. We simply release the light chains with reduction, and then we could analyze these on a high resolution mass spectrometer. So we'll move back to the very original sample, this man from the woods again. So we took normal human serum, and this is before we worked out all the details, and you can see how complicated these spectra can be on normal human serum if you don't have all the chromatography and the ionization right. But this is what normal human serum looked like. Then we spiked in a monoclonal drug to stimulate myeloma. So we spiked in Humira, which is monoclonal. And you can see there's peaks over top this polyclonal background. And when we looked at the man from the woods, he showed none of these peaks. So we were confident that there wasn't anything monoclonal in this patient. So just to give you the outcome of that, well, this patient really had a high rheumatoid factor, and this is a known problem for SPEP, is that rheumatoid factor can glob things up on a gel and you can overread it. He ended up getting a bone marrow because they were still concerned about that abnormal free light chain ratio. No evidence of any plasma cells, but he had a family history of rheumatoid arthritis. And so this guy really had hyperviscosity syndrome secondary to just a high titer rheumatoid factor. So, okay, so now we had a method, we found it, but it still took us with the chromatography 30 minutes, and we still don't know the isotype. So we had to come up with a method that also isotyped for us. And to get rid of this chromatography, we turned to MALDI-TOF mass spectrometry. So we did just exactly the same as traditional IFE. We replaced it with different immunopurifications. We purify the G, the M, the kappa, and lambda. Then we incubate and wash those. We put them on a multi-plate. We do the acquisition of the spot, and then we overlay these spectra. And we had to develop our own software for this. And this is what our software looks like. What we ended up doing, so you can interpret this as these are the lambda purified. So these are all the lambdas that were in the patient's serum. So they could have been attached to a G, an A, and M. They could be free light chains. These are all the kappa in orange. All right, and these are the light chains that were once attached to a G. These are the light chains that were once attached to an A, and these are the light chains that were attached to an IgM. And so what you see is you see the normal kappa lambda distribution for the G, which is 1 to 1.5. Here, A is known to be 1 to 1, which you can see here, and the M, low level of M there. But if you had a monoclonal protein, what you see is you can see a spike that was isolated with the lambda pull down and also with the G pull down, because I don't see any peak associated with the A and the M. And this is how we're making our diagnosis. We call this mass fix in the literature because the hematologist told us they wanted something that had a name similar to Immunofix, and so that's what we called it. So this also allowed us to change the workflow in the lab. So now we've put this into a liquid handler. We have automated spotting, barcoded all throughout, and we're using the same Bruker multi-top that's used in the microbiology lab to do all this. But we had to create our own software to do that, and we created this mass fix software that then coordinates all the workflow through the lab, and really though has sped up the throughput for our lab also. This is what it looks like. I think I'd take you through a typical patient. This is what our software looks like. I showed you these spectra. The patients are up here, and I click on one, it brings me in the spectra. Over here are the diagnosis that was entered by the tech. And over here, because these are very simple files, they're just text files, we can save them in our history locator. And so all the history for the patient is over here also. I've just blocked out the patient names so you can't see them. So you can see a peak here that isolates in the G and a kappa, and you can also see something small here in the A and the lambda. So what we do is we've known that some of these therapeutic monoclonal antibodies that these patients are treated on can interfere. So what we do with those is just simply measure the mass of the drug's light chain. And then we have a little tab up here. You can see we click daratumumab. It puts on here where the mass of daratumumab is. So we're pretty sure that this must be daratumumab. We then see this A lambda, and I can come over here to the history and see that we've been calling A lambda before. But the beauty of using mass as compared to the electrophoretic migration pattern is I can go back to the old patient sample. So this would be the previous sample I saw. You can see this A lambda here. I can mark that mass, and then once I do that, it gets transferred onto the new spectrum. And I can say, yes, that's lining up with the A lambda we've been seeing all along. So I can enter a diagnosis. This is an IgA lambda. It also has a small G kappa was detected with a light chain mass suggested of daratumumab. Now, it doesn't mean it's daratumumab just by the mass. And then we tell them if the patient is not on daratumumab, then this must be a monoclonal gammopathy, but this patient is being treated with daratumumab for their IgA lambda myeloma. So that's how it works out for us in the lab. But if you think it's all about the science, I learned a lot in this this process. You know, building our own software. If you find a good IT person, make sure you treat them nice, because that couldn't have been done without the work of some really sharp IT people to build this software for me. There was a lot of work convincing the hematologist. The Mayo Clinic has been running IFEs here since 1960, so it was a big chance for, a big leap of faith for our hematologists to jump over to this. There was intellectual property rights, licensing that all happened was another big story. But getting paid for the assay was one of the ones that really would drive you crazy. The CPT codes weren't available, so we had to do all this stuff, including getting endorsement from professional societies, gap filling, crosswalks. So if you need a horror story or a long story, you can talk to me about this part of it. And I tell people this is the part that'll drive you crazy. But we finally, that took us alone probably 2 years to get over those hurdles. All right, enough of that part. I want to show you some interesting cases from our go live. We've been live with this assay since 2018. And I talk about increased specificity. So this is a 52-year-old man. He had a positive lambda AL biopsy, a fat biopsy. He came into the lab and we called it G kappa. And of course, you're going to get a call from the hematopathologist saying that doesn't make sense. We're seeing lambda in the amyloid and you're calling a monoclonal G kappa. So we happened to, this was before we were live with the assay, so we were developing it. So I put it on there and you can see a lot more detail in the serum mass spec than you can see off this gel. So here's that G kappa. It's isolated in the G and the kappa, very small. But you also can see a G lambda, a few G lambdas, and then you see this one that doesn't seem paired with a G, and that could be the free lambda that's causing the AL amyloidosis. Well, eventually we did get the urine on that patient, and now you can see in this area there is a restricted lambda on the gel. So there is a lambda present that we could detect in the serum. And then I can run the same mass spec on the urine, And here is that lambda again, isolated here. And because I'm using mass, I can line this up with the serum, and that's what I mean by increased specificity. We also had an interesting opportunity to relook at some screening samples that were done back in the 1990s here at Mayo. Bob Kyle, who really did all the prevalence of Muggus in the Olmsted County cohort, screened almost everybody in Olmsted County over the age of 50 in between 1995 and 2002. And from that, he found 605 patients that were positive, and that made up the Olmsted County cohort. Now remember, he only screened with serum protein electrophoresis, but it's probably a cost issue at that time. But there were all these patients who were negative in Olmsted County who then later over the years came back and had a positive diagnosis of an M protein by IFE. So we were able to dig up from the old 1995 samples their original serum and retest it by IFE and the mass spec method. And what we found is that if we would have used immunofixation back in the 1995s, we'd have added 24 more patients to this screening cohort. But the mass fix assay picked up 50% of those patients. So we knew we had increased sensitivity with this method. Now this shows another advantage of the specificity. This is a 61-year-old male, had IgG kappa myeloma, underwent autologous stem cell transplant 2019. So here's the serum protein electrophoresis prior to treatment. You can see he had a 21 gram per liter M spike. We did the mass spec on that as a follow-up. Came back in 2019 after treatment. We lined this up. We see a 3 gram per deciliter residual amount, lining it up by SPEP. But when we went to the mass spec, what we saw was there was a new IgG kappa that was forming in this patient in the same region. So this gave us specificity too to follow the patient, because we can use the mass as I showed earlier in the patient example. And this becomes a real challenge. Using mass is really important because patients post stem cell transplant, a lot of them look like this as their bone marrow is repopulating with their transplant. You can see all kinds of different clones here as the immune system reestablishes itself in the patient's bone marrow. So if I didn't have the mass, I knew this is where this patient's IgG kappa was. I'm not really suspicious for much here in this mass range. But if I just looked at clones, I could count almost, 21 different clones in this patient. So again, this is how specificity is helping us track these patients. I've already shown you this. One of the challenges for some of these patients is these drugs that they're using to treat myeloma are also monoclonal antibodies. They're all G-kappa at this point. And so differentiating a G-kappa M protein for daratumumab becomes a challenge. But we found mass spec helpful because, as you can see, as this patient gets treated with daratumumab, we can see the daratumumab coming up, but we can still see the patient's M protein. We also know we have outstanding analytical sensitivity. This is a patient, 66-year-old IgA lambda myeloma patient undergoing treatment. When we went live, the patient was already under treatment. So this was 10/2018. The total IgA in this patient is 28 milligrams per deciliter, kind of low. The free light chains are all below the reference level, but we can still see this A lambda in the patient, as I showed you earlier. Then he comes back in a few months later. And so the way we have set our software up is we normalize to the highest intensity peak. So now the IgA has gone down even more. It's down to 6 milligrams per deciliter. The free light chains are undetectable now in this patient. But if I release the restriction of holding everybody to the same intensity standards, I can still see this IgA lambda in the patient's serum at 6 milligrams per deciliter. Right now, our hematologists have asked us not to read this sensitive because it's still controversial whether or not we're treating myeloma patients all the way to 0 MRD. So part of the new trend in myeloma is to think about MRD. And so what people have turned to, since there's not a good serum marker, they've turned to bone marrow, high sensitivity flow or next generation flow, where they're enumerating quite a few plasma cells in the patient's bone marrow biopsies, and they're classifying them as MRD negative or MRD positive. But what we saw was that we are finding some patients who are MRD negative by flow, but yet still had M protein detectable by our new assay. So we lined these patients up and we looked at their progression. So this is a patient who was negative by MRD flow, but negative by our new MassFix assay. But this is a patient who had next generation flow negative, but MassFix positive. And you can see their time to progression is different than those who are not. So being double negative is very helpful for the long-term outcome of these patients. But then, as I always say, when you get new eyes, you see new things. As I said in the beginning of the talk, the reason we chose the light chain is because we didn't think there were many post-translation modifications in them. Then all of a sudden, we kept detecting these patients with M proteins who had very broad mass distributions on the MALDI mass spectrometer. Through a lot of work, we determined that these patients actually are getting glycosylation on their light chain. And some of the early genetics we've done on these patients shows that they're getting glycosylated in areas where the germline would not allow for glycosylation. So we think that some of these glycosylation sites are happening as this antibody goes through somatic hypermutation for affinity matching towards target. So some of these get glycosylation consensus sequences in them. So at first they were sort of annoyance because they're harder to see. It pushes the mass outside of the mass range. But then we started to do some exploring. And the point here is that they've always been there. These are some patients. This patient right here is actually glycosylated. These two aren't. There's really no way to tell whether the light chain is glycosylated off IFE. So we went back to this cohort and we had some inkling that this glycosylation was closer associated with the diagnosis of AL amyloidosis. So we went back to our Olmsted County cohort, redid all the positive patients, found which patients had glycosylated light chains and looked at that progression over time. And sure enough, these patients with glycosylated light chains are at higher risk for ending up with a diagnosis of AL amyloidosis. But to our surprise, that progression to any plasma cell disease, as you can see here, blue is unglycosylated light chain, red is glycosylated light chain, that these patients are somehow developing myeloma and all those plasma cell disorders at a higher rate than patients with unglycosylated light chain. And the mechanism behind that, in my mind, is still much a mystery. But one of the other unexpected findings too was this IgM light chain glycosylation in cold agglutinin disease. Now, cold agglutinin disease is a rare disease. Cold agglutins are quite common in blood banking, but cold agglutinin disease is more rare, where the patient actually has symptoms associated with their cold agglutins. And what we found was, out of the 438 patients that we've found so far with a monoclonal IgM, 14 of those patients in the clinical record had a document cold agglutinin disease, and 9 of the 14 had this light chain glycosylation. So you can see what it does to the MALDI mass spectrometer, or the reading is it pushes it outside. This red box represents the normal mass range. We expect to find light chains, pushes it out and makes it multi-peak because the glycosylation pattern isn't clonal. All right, so with that, I'm coming towards the end of the journey here. I think there's a lot more to do with this assay. It's been quite the journey. We've been live with this assay now since 2018. I just looked today, we've slightly gone over 60,000 samples at this point, and we're pretty pleased with the overall outcome. We are just getting ready to publish our results in terms of the performance, and we've had very low repeat rates, somewhere in the neighborhood of 1.5%, which we think is a little lower than what we had with IFE. We've also not seen any cases yet where serum protein electrophoresis picked something up that we didn't find it on the MALDI mass spectrometer. So we think that, we're coming towards the end of this journey, but there's more journeys to be had. So with that, I think I'd just like to acknowledge some of my coworkers here, and a lot of people did a lot of work, and really I can't thank Dr. Kyle enough. Dr. Kyle had the insight to collect patient samples. We have samples going back into the 1960s here at Mayo Clinic on patients with all these diseases. Without that, we wouldn't have been able to push this assay along. But with that, I'll take questions. This is a good picture from the day we cut the ribbon to start the assay. That's Bob Kyle here. He's really we consider him the father of myeloma here at Mayo Clinic. Mindy Cohagen, who did all the hands-on work to get this assay running. I can tell you when we first got these MALDIs in, and first week I was about ready to think this wasn't going to work. Dave Barnage, a mass spectrometrist who helped me with this. Maria Wilrich, who's my co-director in the lab, spent a lot of time. Angela Dispensary, hematologist, who helped me with all the clinical studies. Surendra, he was the informatics genius. John Mills was my chemistry fellow, who actually did all the work on the mass spec assay. So with that, I'll take questions.
Excellent, Dr. Murray. Thank you so much for the talk. I do have a few questions in the queue. We do have about 10 minutes for them, or let me see. Yeah, about 10 minutes, maybe 4 or 5 for these. The first one is the cost of mass spec, need for dedicated lab personnel, professional staff for interpretation, as well as caseload needed to break even. So I think what they're asking is, how many samples would it take to go through and do what you have done, notwithstanding the differences in PLA codes?
Yeah. So so there's a lot in that question. The cost for us is fairly cheap because of we're using reagents that we classified ourselves that are relatively cheap reagents, right? This technology has been licensed to commercial companies, and I'm sure it won't be as cheap for other people trying to do this as it is for us. That's just the economics behind it. The breakthrough point, yes, I don't think this is something that I would do unless they find a way to multiplex, for bacterial typing or something like that. This would take a significant volume. I haven't done those calculations. I can tell you that our volume's we do about 400 of these a day. So for us, it really pays. The analysis we did saw that we get about a 30% increase in result per FTE because it's just so automated and there's a lot more time for the techs to do it. So it all depends on your situation, whether or not this will work for you or not. Or it will be a financial plus for you or not.
Okay.
I don't know.
There are quite a few other questions. It's hard for me to choose the most appropriate ones, but I'm going to try here. Might we anticipate diabetic patients to have increased glycosylation of M proteins? Any findings? So this is a question, I guess, specifically in folks that have diabetes, and if there's any differences in your assay.
So yes, we know that patients with diabetes do have more of these glycosylated, non-enzymatically glycosylated. The ones I was talking about that were at a risk were not, they were an enzyme put those glyco groups on. But the patients who are diabetic do have higher concentrations of the glycated light chains. Now, whether or not that does anything to the immunoglobulin or not in the long run, I don't know. We haven't done those studies.
Okay, we have some time for a few more, so I'm going to ask a couple. Do you still use IFEE at Mayo?
As of 2 weeks ago, no. We have now taken down, with the exception of doing IgD and IgE, because we just haven't qualified the mass spec for those two because they're kind of rare. So if we see a light chain only disease by our mass spec assay, we then switch it. We then reflect those to traditional IFE with IgD and IgE. That's the only exception, but no other cases are done by IFE. As of 2 weeks ago, we finally switched everything over.
So a timely question, I guess.
Yeah.
Regarding MS for M proteins, since using mass spec for M proteins, since using ratios, does this obviate the need to use internal standards quantitation in the mass spec analysis?
So it could, for if we use an ESI kind of instrument to do this, it could. MALDI-TOF, I don't think so. We've tried to make this internally quantitative and have not really done a very good job. You can kind of get relative numbers, but it's not as good as SPEP. So we are going to make this quantitative in the same way that serum protein electrophoresis is quantitative, except for using total protein. We just measure IgG, total IgG by immunonephrolometry, and then use that to multiply times the spike that we see in the G spectra. And we validated all that, and it looks very good. But the hematologists wanted to go slow. They didn't want to make a big long jump to quantitation until I gave them quantitative data. And so we're getting ready to publish. We've done probably 5,000 cases that we've done comparing SPEP to the mass spec with the outside quantitation with the IgG, IgA, and IgM, and it looks good. And so once our hematologists are comfortable with that, that'll allow us then to get rid of SPEP also. But I know there'll be people who want their SPEP still. So it's
well. So a follow-up question. I think there's 2 or 3 more, but a follow-up question on that particular topic. So you talked about folks not wanting to define presence of minimal residual disease in SPEPs, and it really gets down to what you were saying there with the increased specificity or sensitivity of this method versus maybe an SPEP.
yes.
Thoughts on where that number is going to go and whether they're going to eventually get to defining minimal residual disease?
So right now, everybody wants to know that number for this assay. I've just read some papers recently where other groups are now doing it with the high resolution instrument, not with the MALDI-TOF. And they think that they can get down to the same MRD levels of 10 to the 6 that they're getting with next gen sequencing. I'm a little skeptical of that. I just can't imagine this is more sensitive than next gen sequencing. But then again, plasma cells, that's all they do. They churn out immunoglobulin. So they're sort of amplifying their DNA for us. I made a mistake one time of saying that at a conference, and I think I scared away all the collaborators who were working on next gen sequencing. I said, why pay for next gen sequencing when the plasma cell's doing it itself?
Sort of funny. So last 2 questions. Are there any other post-translational modifications found on an antibody light chain besides glycosylation?
Yeah, there are a few. We haven't published them yet. We're seeing, especially in free light chains, we've seen some cysteinylation. Not sure if it means anything, but it makes the spectra kind of complicated. But cysteinylation is another one we've seen. So we see glycation, glycosylation, and cysteinylation.
Okay, and last one that I'm seeing here, and I'm trying to clean up most of these, is there any differences in software in between, it looks like you derived your own software, but would there be any differences based upon software pathways in terms of the analysis?
No, I mean, the beauty of these MALDI files are just text files, right? They're a text file of intensity versus mass, right? Or M over Z. And once you pull that out, you can then use those and use all kinds of software to plot them in the way you want. Now you could use the software on the Bruker, but it's just not set up for, hundreds of samples a day. It would be a very slow process of trying to overlay all these yourself because it's just not an automated software. I noticed that the instruments that I'm using also have the Biotyper software on it, and also we're just using the traditional software from the MALDI, the Bruker cells. So it could be done. You just have to find a way to make them plot for yourself. You could probably even do it in I don't know. There's probably different ways to do it. We built this for long term. We wanted to be able to do like long-term follow-up and organize it all in databases so we could use it for research also. So that was the idea behind the software we created.
Okay,